Okay,
Fantastic.
so it's lovely to see you all. And without more ado, I'm going to take you through a little bit of a history tour. Next slide, please. Lovely. So a couple of seconds just to explain why I'm talking to you about bioinformatics facilities. So I started out as a wet lab biologist. The only access to computers I had was 2 weeks of introduction to Pascal, mostly on paper, followed by a PhD in cellular immunology in the wet lab, where I was doing a lot of short manual sequencing, and we didn't actually have access to computers. So I went through the fun of doing multiple sequence alignments on squared paper, and I decided that actually there was a better way of doing that. Please, please, please. I then moved to the University of Oxford, where I worked as a wet lab virologist, and I started using computers for my analysis, and I started off being a very frustrated user. Then I started moving on to be a more heavy user of the system. I then started providing support to other people in my institute. I started demonstrating on courses run by the bioinformatics facility, whose resources I was using, and then I went to work for them. And at that point, I had to learn system administration from the ground up because we did everything. We ran our own services. We ran even a large mail server. And I had to learn everything because I didn't really know very much about computers, as well as helping with the bioinformatics and learning to install software and train people to use it. I eventually became the manager of that bioinformatics core. And then I had to learn a lot about finances and planning and user accounting. And then I was very lucky because I had the opportunity in 2002 to start up and run a new bioinformatics core facility in Imperial College in London. So I had the opportunity to discover how to try and go around some of the mistakes that I'd inherited or had managed to add to in my previous role. And I ran that facility for 16 years until I moved to the EBI about 2 years ago, where I now run a software development team. So a lot less bioinformatics, a lot more data, and a lot more infrastructure. Next slide, please. So what I wanted to do very quickly is give you a very fast tour through bioinformatics facilities that I've known since the 1990s moving forward to the present day, and then use that information to try and look at where the similarities are in terms of what I feel a bioinformatics facility does, and then to move through how the role is changing over time and how we can make the best use of our resources to help researchers. So to start with, some bioinformatics core facilities actually predate the regular use of the word bioinformatics. Some of them started out a very long time ago. As an example, in Oxford, UK, there was a molecular biology data centre which started as a means of sharing protein data from the PIR database, if some of you remember that, by being able to run local copies on tape. So what was around at that time? Well, there was no human genome sequenced. There weren't any microarrays. They hadn't been invented. There wasn't any Unix on the big servers. Wasn't really in use. And there wasn't very much going on in the World Wide Web either. Most computers were not found in labs. If you were very lucky, your institution might have a dumb terminal to access a VAX or a VAX cluster. There were a few general use bioinformatics programs around, and you might be able, if you spent a lot of money on a commercial system, to be able to do some basic protein modeling using PDB coordinates. Next slide, please. So moving on in time, we're now in the early 1990s, and this is around the time that whole bacterial genomes started being published. These were a major effort. They took large consortia many years to put together, and a small rash of those first came out in around 1995. The Human Genome Project has already taken off, and looking at what was around in institutions, You might find that there would be a gel-based sequencer, or maybe a couple of gel-based sequencers kicking around, and there might be a team offering oligo or peptide synthesis, but there weren't that many core bioinformatics or other bio-related facilities. Looking at the size of data, well, the EMBL database fitted on a CD-ROM. There was still quite a lot of different bioinformatics programs kicking around, and you could look on the World Wide Web and there'd be some interactive content on some sites. And we started to see the start of national bioinformatics nodes and networks being set up. But you couldn't go out and employ a bioinformatician as such. There weren't really any formal bioinformatics qualifications. Bioinformaticians tended to be biologists who programmed. And if we look at the sort of hardware that a bioinformatics facility might be running on, if we take Oxford as a case in point, they had a VAX cluster, which then moved to a single processor Unix server with about 1 gigabyte of disk space, and that was serving the entire university for all of the biologists that wanted to use it. Next slide, please. So moving on to the late 1990s, this was the time of heavy use of the draft human genome. So there were various ways of viewing that. Ensemble and the UCSC browser were very familiar to many. EST clustering was very important if you were looking for your favorite gene in the draft genome. So some big teams were sequencing and annotating their own bacterial genomes, but these were still big team projects. They were still taking quite a long time to do and required quite a lot of technical competence to actually put together a bacterial genome. This era was the start of some very heavily used open source bioinformatics projects such as EMBOS. And we're seeing at this point that many labs, many offices had their own desktops, so people were becoming generally more computer savvy. You probably weren't going to have to teach people to use a mouse anymore. And looking at the data, many core facilities were pulling down and locally indexing the main public databases. So you might find a local copy of the whole of EMBL, the whole of Tremble, SwissProt, and various other databases, all indexed locally so that your users could blast search against them and interrogate the data to find sequences of interest. And mainly because of this, the amount of local storage used by core facilities was growing, and the amount of compute that they required to actually do the work was also growing considerably. Next slide, please. Then we come to the early 2000s, and at this point there was what I would call a disruptive technology change, because the microarray came along. And this was really the era of the start of standards, with the Miami standard for the minimum information about a microarray experiment. So microarrays had a big effect on many areas of biology. And around this time, very often, if you look around institutions and larger universities, you would find that they would have maybe a microarray core facility, or a bioinformatics and microarray core facilities, and very often there would be some sort of local sequencing available as well. So around this time, the local copies of the large biological databases like EMBL and GenBank became too large for most institutions to want to keep their own local copy. But that was okay, because they were widely available on larger sites, along with simple web interfaces for many local maintained programs. And at this time also, we were starting to see the start of distributed computing, so-called e-science or grid computing, being used to share distributed compute across multiple sites. So this started off as a research area in its own right, and eventually turned into what we would consider to be the cloud today. Next slide, please. And then we have a disruptive data event. This was the arrival of the draft human genome in the public databases, and of course the seminal Nature and Science publications of the human genome. And I just want to draw your attention to the pie chart for a second. So this is what the EMBL database looked like around the time the first draft human genome appeared in it. And at that point, over 60% of all of the information in the EMBL and of course the NCBI's version of the database is all human. And there were a lot of questions then, what on earth is going to happen to all the people that work on plants and bacteria and fish? You know, everything's gone human, what's going on? And then of course, the people that were working on human data were suddenly discovering that the draft wasn't very stable. Your gene might appear in one release and become twice the length or half the length or on a different chromosome in the next release. So how did you make use of that data? The questions came in very thick and fast. Next slide, please. Moving on from the late 2000s onwards, well, the main databases are too large to index and store locally for almost everyone. So many researchers were now using centralized, very rich web resources and web sites and web services to enable them to do bioinformatics on those sequences remotely rather than relying on large local compute. This is the era of the rise of systems biology, and this taught us some very important lessons. If you want to do proper data integration across different data types and you want to model, this really highlights the need for very good metadata. Otherwise, you can't do meaningful data integration. And this led to the proliferation of many, many, many new minimum standards. All of those lovely minimum standards that you had to be very familiar with if you were going to submit your data to any public repositories. And around this time, many researchers were now trying multiple technologies, multiple types of omics. And very often these were research groups that didn't have a background in those data types and those types of analysis. And so they were looking for more and more help. And then around 2006, we had another disruptive data event. This is actually a technology event as well, because this is around the rise of the next generation sequencing technologies that were coming in. And of course, these were quite unstable to start with, and we had many different competing platforms. And this led to a very rapidly changing set of data formats and the software used to analyze them. And we'll look a little bit later on how these disruptive technology events influence what a bioinformatics facility needs to do to make the best use of this new technology. Next slide, please. So I wanted to draw your attention to a quote that I always thought was a very interesting one. So back in 2003 at the O'Reilly Bioinformatics Technology Conference, Lincoln Stein was giving a very interesting talk, and he actually said, and I quote here, "The field of bioinformatics will be gone by 2012. The field will be doing the same thing, but it won't be considered a field." Now, I just want to reflect on that for a moment. So we're all doing bioinformatics. That's pretty much why we're here. But I think he's kind of right, because bioinformatics has become so ubiquitous to biological and biomedical research that it's really viewed very much in the way that we now consider molecular biology as a technology. It's there, we all do it, and actually it is still a field if you're a bioinformatician, but it's really grown. It's become part of what most biologists have to handle on a daily basis. Next slide, please. So that was a very galloping history lesson through a number of years. So I think it's useful to look at what the basics of a bioinformatics core facility are and reflect on the fact that we're all doing a good job. There are many, many different bioinformatics facilities around. And if we all stopped and talked to each other, which we will do in a little while, about what our facilities do and what we offer and how we're funded, we will find that we're all in the business of bioinformatics, but we all handle the core facilities in slightly different ways. So let's just stop and look at what the basics of a core facility consist of. Next slide, please. So these are the goals that I consider to be key for a bioinformatics facility: to provide bioinformatics users with what they need to do their research, basically, infrastructure, training, and support. But we don't just support bioinformatics users, we're probably also supporting bioinformaticians and other specialist users. So those could be writers of software, they could be statisticians, and they need support as well. So that can be the infrastructure they need to drive the tool development and the technique development that they do. And then in order to do that, a core facility needs to acquire, maintain, and develop the infrastructure and the trained personnel that are required to do the job, to provide these facilities for our users, be they biological researchers or bioinformaticians or statisticians. So what do we actually need to do that? And how is it changing over time? Next slide, please. So I just wanted to put this slide in because this is something that comes up in discussions a lot. Does a core facility provide support to users, or does it provide service? So what's the difference? So bioinformatics support, or any type of support, is helping other people to do their job, to get their job done. How you provide help could be by training them to understand their own data and to analyze it. It could be mentoring them. Let's look at our data together. Let's have a look at it. What do you think of the quality here? Let's try doing this. It could be helping them, showing them how to use a program, setting up their storage for them. Service, on the other hand, is basically doing the job for them and generally getting paid to do it in some way or another. So many bioinformatics core facilities use both approaches. Some of you probably only using one. There's a continuing requirement for both types of approach, and sometimes it's quite difficult to make the definition between the two, because an individual project might require both support in some cases and service in another. Next slide, please. So I wanted to go back to quite a long time ago, when we had a big discussion group of international bioinformatics core facility people discussing what their challenges were. And this is a summary that I prepared after that meeting in 2003. So the key challenges for a bioinformatics core were to provide stable infrastructure. So infrastructure was going to be there long enough to actually make a difference to a lot of projects, and to have a knowledge base that was present over a long period of time. But at the same time, to be able to stay flexible enough that resources could be reallocated as demand changed over time, to stay in touch with their users and their funding agencies to make sure that they stayed aligned with what their users needed from them, and to find the right staff, to retain them, develop them, and to reward them so that they stayed there for a long time and were available to build the service over a long period. So the key challenges for funding were to find a business model that provided both stability and allowed the service to grow when needed, and to find out a way of charging that people would actually pay. So I would imagine that a lot of those are resonating with yourselves and probably sound very familiar. Next slide, please. So, excuse me. I looked at the areas of work that were outlined in 2003, being used by many core facilities. And then I went through them and I added how things have changed in 2019. So some areas have stopped being particularly important to bioinformatics facilities, or certainly the ones that I was familiar with. As an example, we weren't really supporting gel-based sequences anymore in 2019. But almost all of the things that we were doing in 2003 was still being used in 2019. Keeping very large copies of public databases, not so much, they're just too big. But subsets, yes, and hosting secure data sets, yes. But mostly these were growth areas. The facilities that I was working with in 2019 were often running services that simply didn't exist in 2003. So I've starred what I saw as the growth areas, but in particular things like grant writing. In 2003, we weren't really directly involved in writing grants. By 2019, this was a very large part of what we were doing, also holding grants and writing our own. We were involved in writing more specialist software. We had a lot of core facilities, so we were involved in laboratory information management system development, LIMS development, sample tracking software. But we were also quite heavily involved in advocacy, brokering, skills sharing, collaboration, and supporting other bioinformaticians and other core facilities. And I'm going to talk about that more in just a moment. Next slide, please. So a lot of the questions that were being asked by users about data in 2003 were still very much there and now. The sort of questions that you probably hear on a fairly regular basis. How do I store my data? I've got loads of it left over from this project. My PhD students just left. How much of this data do I need to keep? Do I need to keep all of my interim data? What can I throw away safely? I'm not sure about it, so maybe we need to keep everything forever. Oh, but I've got nowhere to store it, so can you back it up for me? And I've now got a huge sprawling set of directories. How do I find the vital bits of my data? And how do I compare it with other data sets, public and private? How do I ask meaningful questions about my data? How do I integrate my mass spec data with my NMR data, with my microarray data? How do I share my data with other people in the group? And what do I publish? What format do I use and where do I send it? I think if we look at these types of questions now, they're all still there, but the data volumes are much larger. The number of data types that any biological researcher has to deal with are much larger in general. We're working with different tools to analyze and store our data, but there's a much higher expectation generally about data integration and being able to find the information that people require. And then on top of that, I think most researchers are a lot more savvy about the fact that some data are sensitive and there are general security worries, particularly around human-related data. Next slide, please. So it's not really just about running BLAST anymore in the average core facility, if there is such a thing. Next slide, please. So one thing that I see as a very important growth area for bioinformatics facilities is adding value via using our information to help researchers find each other and share information. Because the types of data that most biological labs are generating now is much more complex and heterogeneous than it's ever been before. And very often research groups are wanting to try methods that they don't themselves know very much about. And in fact, you may find that a very large lab will want to use techniques that perhaps not even everyone in their own department has the necessary skills to understand and work with. So matchmaking skill sets is a really important part of what a core facility can do, because if people come to you for help, you'll have a pretty good idea of what their general skills are, and being able to keep people in touch with each other and help them to find other people and other facilities that have the information they need to try out new methods and new techniques is really, really helpful and can really speed up research. If you can also be involved in more general focus areas for good practice, that's also really, really important. So if you can share your knowledge on how to make the best use of metadata, how to publish your data appropriately and get it ready for publication using the right minimum data standards, that's the sort of information that can help a lot of researchers very quickly. You're in the position to share that best practice, to produce good quality data and make sure people know how to value that and how to use it. And of course, you can also provide domain-specific expertise for other groups. Maybe your institution has mathematicians who want to do some modeling, but don't know how to pull raw data out of a particular database. They may need data set cleansing because they may need somebody locally who understands the biology of the data set that they're trying to analyze. And sometimes having a bioinformatician around that can mediate between somebody running a vanilla HPC setup and a biologist that's trying to use it can save everyone a huge amount of time and effort. Next slide, please.
I just wanted to quickly say you have
Please.
5 minutes left.
Yep, got you. Thanks. So integration, being glue, bridging the gap between people that need to work together but don't necessarily have all of the information to hand to share their information effectively is really important. And we learned this very much when systems biology took off. You can put biologists and mathematicians in a building together and tell them to do good science, but you need the glue. And the glue might be the bioinformatics core, because the chances are you will understand the biological data, you'll understand some programming, you'll understand the data types and some of the methodologies that we use to generate the data. So you'll probably have a pretty good idea about where the errors are coming from as well. And this sort of information can have a huge effect on the type of research that's carried out. Next slide, please. So now we tend to find that many institutions have multiple core facilities that are used by the bio or life sciences health community. Working with other core facilities becomes a very important part of the bioinformatics core. Excuse me, because all of these facilities are going to have one thing in common, and that is the data life cycle: data generation, data cleansing, data analysis, data storage, data submission, data sharing. And very often they're going to be sharing clientele as well, because somebody that gets their sequencing done in a sequencing facility is probably going to need somewhere to store that data and some help with analyzing it. And that's just at a very trivial level. So a bioinformatics facility can really help make sure there's smoothing between all the areas of the data lifecycle. Work with your other core facilities, talk to them, understand what they need, help make it easier to move data from one facility to another. And you can provide services for other cores as well. That might be statistics to help with experimental design before the experiment is done. It could be data management or offering physical storage. And there's always opportunity for outreach. And sometimes working together with other core facilities can have a huge effect on your funding. If you all work together, there are generally more funding opportunities. Next slide, please. Now, a long time ago, there weren't very many bioinformaticians around. There tended to be research teams that were bioinformatics research based, and there tended to be wet lab research. And very few wet lab research teams had their own bioinformaticians. That's very much changed over time. Many biologists have some bioinformatics expertise. Some larger teams have their own embedded bioinformaticians. They might have statisticians and sometimes software developers as well. So how do bioinformatics core work with those people? Well, it's very nice if we can provide infrastructure services for them and facilitate their access. If you've got software developers building local software, you can help to disseminate their software and interfaces and tell people about them. And it's important to do gap filling too, because a single bioinformatician is not going to know everything. We all have our own areas of expertise. So if we can network with those people and encourage shared knowledge, encourage them to come and help teach and share their information, teach them, offer them support. And also, of course, you can help with recruitment. You can add your shared knowledge to the researchers that are trying to recruit bioinformaticians to make sure that they know what they're looking for. Next slide. And research software development's really taken off as well. As we look going backwards, it's always been part of a core facility remit, but it's got a lot more important in many cases, and it's become much more of a task in its own right, making software that is actually significant software that's maintainable, reusable, properly shared, and providing automation, because all of the things that we do are larger and faster, and data are generated much more rapidly now. So providing automation is much more important. And also providing infrastructure for other developers is also important. If you've got software developers writing software that needs to be Dockerized, providing virtual machines as sandboxes, providing containers, helping with continuous integration pathways, all of that can have a huge effect on the ability of other people to provide research software development. So these are all just areas I'm touching on that are real growth areas that can really become a very important part of what a core facility can do. Next slide, please. We're coming to very near the end now. So training and teaching has always been an important part of a core facility, but how we do it and the subject matter we're teaching is obviously changing over time. There's much more emphasis on remote learning, e-learning, learning via Zoom, and it gives a much more flexible timing for wet lab researchers. And there's a much bigger body of commonly shared resources for teaching than there used to be, and it's really, really nice to be able to share resources and see how somebody else ran an RNA-seq course, and if you want to illustrate a particular point, has somebody else already published a data set that shows exactly what you're trying to show. It's all great stuff. Courses are still really, really important, but as the data sets get bigger, of course, you have to be much more careful about how you tailor the data sets you teach on to enable the pipeline you're showing to actually run in the timeline that the people on your course can actually see it. And of course, another point is if you're running out of time because you're busy, writing and producing training courses becomes usually the thing that gets pushed to the back of the queue. Next slide, please. So what happens when a new disruptive technology comes in? Well, generally, and this is all very highly generalized, the ability to generate, for a biologist to generate a new data type using a new technology tends to appear faster than the tools that are needed to analyze that new type of data from that new technology. And also, on top of that, the size of the data that are being generated and the number of tools in the toolbox needed to analyze that both tend to grow faster than the knowledge in the general user community that's needed to actually use those tools and make the best use of those data. So why is that important to a core facility? Next slide. Thank you. Well, we're here to address the gaps. So when a new technology comes in, the chances are somebody's going to come through your door going, I really, really, really need to use this technology. Can you help? And in order to help, then the core facility has to itself learn how to use that new technology, how to make the best use of the data coming out of that technology, how to understand the errors in it. And very often that also requires installing buggy software, early analysis software. Maybe it doesn't compile, or maybe there's nothing there to do what you need to do and you need to write some yourself. So what are you doing? You're plugging that technology gap to help other users make the best use of that new technology quickly. And part of that is a critical evaluation of everything to do with that technology, how the data are generated, where the errors are coming from, how to do the best type of analysis to make the best use of that data. Once you've found the good practice, you can share it, you can document it, you can teach other people to do it, and you can install what is needed locally so that other researchers in your institution can do it better and faster next time. And that's really, really important. Next slide, thank you. So we don't know what the next disruptive technology is going to be for bioinformatics or biology or life sciences or healthcare data. It might be sequence-based, like the last one was with next generation sequencing. It might be based around confocal microscopy. It might be based around new types of mass spec, or it might be single cellular level multi-omics. We don't know. I mean, there's lots of different people would put money on in the bar on which one it's going to be, but we know it's coming. And next slide. Thank you. What we do know is that whatever that new disruptive technology is going to be, our researchers are going to want to use it. The majority of them are going to need some help getting to use the new technology and making the best use of it. And there will be challenges in learning to use next technologies as they come through, because there always are. And we know that bioinformatics cores are going to be in the thick of that, making those technologies work for researchers so that they can just embrace that technology and get on with their science. And of course, somewhere in a bioinformatics facility near you, somebody's going to be writing a new course on it. So thank you very much.